Hello, my lovely anatomists and physiologists. We are ready to talk about the homeostatic regulations for the cardiovascular system. And when we talk about homeostatic regulations, we're talking about mechanisms of control for blood pressure. We're talking about homeostatic mechanisms for blood flow. And we're talking about homeostatic mechanisms for perfusion, where perfusion, remember, is talking about movement of blood to your tissues. And so we can think here to like distribution. Remember, we don't really have enough blood moving through the body to equally deliver to all of our cells. And we don't need that, right? Because not all of our cells have equal need, right? Oh, we can really think about cardiovascular system as a model of equity, right? Where equity is different from equality. Equality is this idea that like everybody has a pair of shoes, whereas equity is the idea that everybody has a pair of shoes that actually fits their feet, right? And so when we talk about blood flow, of course, the job of the cardiovascular system is to make sure at minimum, all of our tissues stay alive. So they need enough oxygen, enough nutrients, enough waste removal to stay alive. But, as we're doing different stuff in our body, we're going to have different levels of need throughout. Now, that's not really true for the brain. So we do see that the brain gets like regular blood flow. So it's always staying nourished. Okay. When we talk about these homeostatic mechanisms for blood pressure, blood flow, and perfusion, what we'll see is that we can get neural control, which we've actually talked about. So we already talked about the cardio acceleratory center at the medulla oblongata and the cardio inhibitory center also at the medulla oblongata. And then don't forget, we have vasomotor control also it's the vasomotor center also at the medulla oblongata, right? And then of course we're seeing that those signals are traveling out through either the sympathetic or parasympathetic nervous system. To pick up on our details, we're also going to see autoregulation, which we've talked about kind of a little bit throughout our videos, but we're going to summarize and bring that information together in one place. And then we have not really talked about the endocrine regulation of the cardiovascular system, so we're going to be doing that as well. Let's go ahead and talk next about the autoregulation, and then endocrine needs its own video. So we'll be doing that toward the very end. When we talk about autoregulation, we should think of very much like local receptors. Local receptors, and we're going to see in large part that these are going to be like chemoreceptors. When we talk about the local receptors, we're very much talking about like a self-regulatory system or mechanism. And I think we should consider there's like two areas. So I think we should consider we have like this regulation on our arterioles. And then we have dysregulation on our precapillary sphincters. And just to kind of keep the context, the arteriole comes first, before the precapillary sphincter. When we were talking about this kind of autoregulation as it relates to the arteriole, we're going to think about this as a myogenic response. Myogenic response. And I think probably the key takeaway here is the arteriole is feeding into the precapillary sphincter. So we're going from a small tube to a super tiny tube. And it's the job of the arteriole to kind of make sure the capillary can handle it. So we could consider, like, it's the job of the arteriole to make sure the capillary can handle it. You don't want a big flood of blood going into a super tiny capillary and busting open that capillary. And then you're going to have organ damage, you're going to have tissue damage. It's going to be internal bleeding. Yeah. So it's kind of the job of the arteriole to make sure we're not like super flooding the capillary. Right. And then also we can think about, like, at the same time, we need to make sure we don't starve the capillary. We need to have some blood moving there, if at all possible, right? So what we'll see with arteriole is that it's detected by stretch receptors of the smooth muscle. So this is not a chemoreceptor, this is a stretch receptor of the smooth muscle of the arterial wall. Right? And what we're going to see happening here is, again, let's think of it as stabilizing blood flow to the capillary. Meaning, again, like, we don't want to flood it and break it open, but we also don't want to dry it out. Yeah? Okay, so what we're going to see happening is, if you have low blood flow, so the stretch receptors are not very stimulated. If you have low blood flow, now we're risking, like, not having any blood to go to the capillary. We're risking like a dry riverbed, right? And so we're going to see a vasodilation of the arteriole in order to increase blood flow to the capillary. Yeah. All right. If we have high blood flow in that arteriole, now we're worried about flooding and bursting those little tiny baby capillaries. And so now we're going to see the arteriole will do vasoconstriction in order to reduce blood flow. So here, if we think vasodilation of the arteriole is going to increase blood flow to the capillary, whereas vasoconstriction of the arteriole is going to reduce blood flow to the capillary. Okay. When we talk about those precapillary sphincters, that's part of our capillary bed. So we had a whole video on that. We're going to see here the chemoreceptor is going to be super involved in this case. And so we're going to be detecting concentrations of oxygen gas, carbon dioxide gas. We're going to be looking for lactic acid or hydrogen ions. So these are both indicators of like a lower pH. We're going to be looking at potassium levels. We're going to be looking at histamine, which is our inflammatory chemical. We're going to be looking at prostaglandins. We're going to be seeing endothelin involved here. We're going to see nitric oxide, which is abbreviated NO here. And then we're going to get chemicals from platelets and white blood cells or leukocytes here as well. Our precapillary sphincter, remember, is like this master resource allocator. It's really allowing for the equity in the body, right? It's making sure that the blood is flowing through the capillary bed as needed. So in a lot of cases, it's not needed and it's off, right? And then in other cases, it's needed and so it's open. So what we see, I'm going to say precapillary sphincter as closed and precapillary sphincter as open, right? If it's opening up a precapillary sphincter, you could also think about this as a vasodilator. If we're like keeping the precapillary sphincter closed, you can think about that as a vasoconstrictor. Okay. If we are going to dilate, open the precapillary sphincter and allow fluid, allow blood to move into the capillary, that's happening when you have low oxygen levels, meaning that tissue is really active and it's used a lot of oxygen. That's happening when you have high carbon dioxide gas levels, meaning the tissue is really active, it's produced a lot of CO2 during cellular respiration. As part of cellular respiration, you might also see elevated hydrogen ion levels. You might see in your skeletal muscle activity elevated lactic acid levels to indicate a lot of activity. Okay. We might get elevated potassium to indicate here that our tissue is active. If our tissue is metabolically active, we're going to have an elevated temperature, just localized in this area. So that's going to be an indicator we need more blood flow there. Okay. We'll see endothelial cells releasing nitric oxide as a dilator. So if your nitric oxide levels are high, yeah, we'll see histamine, which is triggering inflammation. Inflammation is an increase of blood flow to a particular area. So obviously we need to open up that precapillary sphincter in order to have inflammation. That makes perfect sense, right? So in each of these cases, we're going to see that these are going to be basically chemical signals, or in the case of the temperature, receptor that will trigger the opening of the precapillary sphincter. So now we are increasing blood flow to that particular group of tissues, to that particular group of cells. On the reverse side of that, we're going to see those precapillary sphincters stay closed or close up, you know, when all these factors are the opposite. So that's pretty obvious, right? So I'm going to say like opposite to open. Pretty basic, right? I mean, you could put in your notes like increase oxygen level, decrease CO2, decrease hydrogen ion, and so on and so forth. In addition to being like opposite, we're going to see some specific vasoconstrictor chemicals. So we'll see prostaglandins trigger closing. We'll see platelet factors. Think about it. If you have platelet factors being released, you're forming a blood clot. So you probably have damage to your vessel. Prostaglandins are interpreted as pain. Again, you have damage to your tissue. You probably have damage to your vessels. So you need to close off that capillary bed just to reduce blood loss at this point. We're going to see factors coming from leukocytes or white blood cell factors. Again, if they're showing up on the scene, you have damage. You have possible infection there. And then remember, those endothelial cells are releasing the endothelin as part of that vascular phase of hemostasis, and this is a strong vasoconstrictor, right? So it's going to help make sure that these capillary beds stay closed. So that's helping to reduce blood flow and blood loss in this particular area. All right. This is pretty amazing and cool how you get this very, very equitable sort of distribution of blood to your tissues on an as needed basis. Okay, up next is looking at the endocrine regulation. So stay tuned for that. And as always, take care of yourselves and each other.